Well, thanks for the opportunity to speak to you this morning. Like all the other speakers, I want to thank the NAK and the program committee and Phil Martin, for inviting me here. I also want to thank my sponsors from last year. I'm a second-year fellow, Bo Fernhall and Wojtek. Thank you very much for nominating me for fellowship status, as well as, most importantly, paying for the meal last night, the $250 bar tab for 5 people. What the hell did you buy, Bo? Bo likes good wine. I'm going to take all political decision-making out of it and stand over here. One, I don't want to turn my back on anybody, and two, I'm originally from a blue state, Massachusetts. I grew up in Massachusetts. The only state, by the way, to vote for McGovern in 1972. And I know Patty's probably sick of me telling this story, but I was an undergrad at UMass, and Patty was the teacher of my first advanced exercise physiology class. And I'd like to think that she had some influence in my career development. And so thank you, Patty, for that class and opening my eyes to exercise physiology. So today, this is a bit different talk than I usually give. I'm usually focused in my data, so Phil asked me to kind of give an overview of the field of exercise immunology. So I'm going to talk about kind of a brief introduction to the field, very briefly talk about, and not doing any justice probably, but Immunology 101, and then talk what I think are 3 observations that have driven the field thus far. And you might see that they kind of bleed out into different areas and different things, and I hope to tell a story, like a historical story, relative to our field, and certainly point out the limitations and the strengths of some of the approaches that have been utilized, and then try to summarize it all up. So, yes, Virginia, there is a field called exercise immunology. The birth of what I call modern exercise immunology occurred in probably the mid-1980s, really about the time that I was going to graduate school. I went to Springfield College for my master's, and then I went to the University of South Carolina at Columbia, where I trained with Mark Davis and Russ Pate, and I established a collaboration there between our department, exercise science at the time, and the Department of Microbiology and Immunology at the medical school, which is about 4 miles away. And I was one of the first, I think, students to kind of bring that to the University of South Carolina, and that's kind of perpetuated itself over the course of time. About 95% of all the publications found in a search of exercise and immunity have been since around mid-1980s, late 1980s. And believe it or not, there's even an international society devoted to this topic called ISEI, International Society for Exercise Immunology, of which I'm a past president. This was founded in 1989 in Germany. It's really neat because we have a meeting every other year, and there's a lot of people who study exercise immunology outside of our country, so I get it, it's really given me an opportunity to travel the world and meet interesting people from different countries. We even have our own journal called Exercise Immunology Review, which has, as most review journals have, a high impact factor. In fact, one of the articles as I was preparing for this, I remember, it's amazing how quickly you forget, but 3 years ago I was an author on this summary article, consensus statement about immune function and exercise, and it kind of dawned on me, oh my good, I should go read that article and then use that to prepare for this talk, and I did, and that was very useful. So there's been really an explosion of exercise immunology research driven by a couple things. The more we learn about biological systems, the more we understand that the immune system is inextricably linked to proper functioning of those systems. And it's really been kind of a surprise and a revelation over the course of my career that a lot of people who do exercise and study various things like neurodegeneration or muscle or obesity and diabetes kind of get drawn into immunology, because immunology, the immune system, is at the pathophysiology and the health of a lot of tissues. And so the immune system can be found in fat cells, it can be found in bone, it can be found all over the place. And so really that has led me to the inevitable conclusion that it's really pretty cool to be an exercise immunologist. Growing up as a child of the 70s, I watched a lot of TV, and probably you did too if you were in the 70s. And so other influences besides Patty that had impact on my career were I like science fiction movies. And one movie in particular I remember watching and being intrigued with as a child was this movie called The Fantastic Voyage. I don't know if you recall those movies. Yeah, those are good. Yeah. See some head shaking, right? And so this was a movie where some scientists were shrunk down, and it had a spaceship that was shrunk down. They injected it into the bloodstream of this diplomat who was fighting for his life, and they were trying to solve the problem. But the problem was that the immune system attacked these guys as foreign invaders. And so they had to battle the immune system throughout the course of the movie, which was pretty cool, to me at least. And then, of course, well, who'd ever think in scientific talk you'd see John Travolta? But here he is, right? You remember this movie, The Boy in the Plastic Bubble? Later, this movie was also kind of at the time where I was watching a lot of TV and movies, and this guy had severe combined immunodeficiency syndrome, which is a genetic disorder where it lacks T cells, and of course that makes that person, very, very susceptible to infectious agents. And T cells were kind of the target of this genetic disease, and this was in '76, and just about in 1980, about 4 years later, that's when HIV started to become discovered, and that was a disease associated with the infection of CD4 T cells. Their depletion then led to immunosusceptibility to various diseases. There's another corollary field that I'm involved in relative to immunology called psychoneuroimmunology. In fact, I did my postdoc at University of Minnesota in the field of PNI. And PNI is kind of a broader field where the questions really relate to the bidirectional communication between the immune and the central nervous system. And so I won't go through this diagram, but I use this in a class that I teach called stress immunology, really as kind of the fundamental communication pathways where your central nervous system talks to your immune system, and conversely, products of your immune system called cytokines can then feed back and affect things like autonomic nervous system output, endocrine output, and importantly, behaviors. I mean, everybody in here has been sick at one time, and you adopt a certain behavioral strategy when you're sick. You're lethargic, you reduce your food intake, things like that. Those are all strategies to enhance survival. And I could give a whole lecture, and I have given whole lectures on that before. So let's talk about characteristics of a healthy immune system. So first of all, your immune system should attack non-self, right, rather than self. We know if you attack self, come on in. Come on in. It's okay. If you attack self, that's known as autoimmune disease, right? So we have to have a way to recognize non-self and self. It should have specificity, and it does. What the immune system recognizes are called antigens. These are small molecular patterns that are recognized by counter-receptors, either fixed to cells like T cells, or soluble proteins like antibodies that then can recognize these antigens, help you to clear them, recognize them, and deal with them. The immune system has a memory function. You all know this because of the vaccinations that you receive, right? If you receive the vaccine, you generate memory, and then when you get infected with a live pathogen, it either doesn't cause pathology or the pathology is greatly reduced. As we'll see, it's located in strategic places. It's highly mobile and diffuse, and importantly, it's highly regulated. You don't want too much immunity, and you don't want too little. Physical activity and exercise have been shown to affect cell numbers, birth, death, and turnover of cells in the immune system, cell movement, and their location. These immune cells traffic in a very specific way that I'm going to talk about. There's been studies that have shown that exercise affects cell phenotype and function, the kinetics of immune responses, and also immune organ structure and function. So it does have a wide range of effects. And this can occur through many different exercise-induced changes, such as changes in body temperature, hormonal signals, neuroregulation, among other things. So there's a lot of things that regulate the immune system that are affected by exercise. So just a little bit about the birth, life, and death of leukocytes, or immune cells. All immune cells are born in the bone marrow. Some differentiate there and mature there and are released as mature cells. T cells, or precursors to T lymphocytes, have to travel to the thymus. That's why they're called T cells, where they are educated. They differentiate and mature and then get dumped into the blood to go out to the tissues. In the blood, some cells are still maturing. Importantly, it's in the blood that these cells transit to various tissues. They can exit the blood through high endothelial cells in the venous circulation where there's sluggish blood flow. So cells can attach to the endothelial cells and then go through a process known as diapedesis, where they squeeze through the endothelial cells and get into the tissues where they can do important functions. So they can travel around in the interstitial fluid. They can still differentiate and mature. And a lot of cells of the immune system really don't undergo final differentiation until they get to the end tissue, where the signals, the microenvironment of the end tissue, influences those cells to take on certain properties which are important to the function of that tissue. And those properties can change depending on the state of the tissue. If it's an inflamed state or if you're recovering from inflammation, the cells of the immune system change in phenotype. And then, of course, these immune cells can drain out the lymphatic system and ultimately recirculate into the blood and go through this whole process over and over again. And so the goal of the immune system really is of immunosurveillance, so scanning for things that are abnormal, for instance, virally infected cells, microbial contamination, bacteria, things like that. They are also intimately involved in tissue repair. So in addition to recognizing pathogens and destroying them, sometimes the destruction phase leads to host tissue damage, and that needs to be repaired. So the immune system also adopts a strategy to help in the recovery phase. Of course, this is very important in exercise physiology and muscle and all kinds of other tissues. And importantly, these immune cells communicate with one another and with other cells, and with, like I showed you in that diagram with all those arrows, with the central nervous system and the autonomic regulatory systems. Cells die through either apoptosis or necrosis. So apoptosis is kind of a clean cell death that doesn't result in inflammation, whereas necrosis is kind of a situation where the intracellular contents become extracellular, and that's really a danger signal for our immune systems to go in there and help clean up that debris, inflammatory response, and then help to resolve the problem and regenerate the tissue. We've got some organs. Even though the immune system is diffuse, there are some organs where immune cells like to reside. These are critical niches where through cell-cell contact, your immune cells are educated to perform specific tasks. And you can kind of look through here some of these organs, tonsils and lymph nodes and the thymus I mentioned, and the bone marrow and the spleen. And of course, if you look at these in this person, you could imagine that there's a challenge in studying human immunology because it'd be difficult to get access to these tissues. So the blood really is very accessible, and we do a lot of studies in the blood, but there, as I'll point out, there's some limitations to just studying the blood relative to trying to figure out what's going on with the immune system. So a properly functioning immune system is dependent on the strength and the type of response. So you all know that too much immune function, too much of an immune response is bad. In the situation of autoimmunity, right? You can have inappropriate tissue damage by having too much inflammation. Allergies is an example of an overactive immune system. You can get fibrosis and granulomas where there's chronic inflammation in tissues that cause tissue dysfunction, and even death. If you're septic in the hospital, obviously you're getting killed by the sepsis and the microbial contamination, but it's really the response to the microbes that's causing the problem, the hypotensive response that doctors can't recover. It's the cytokines commuting with your vasculature, with your brain, that's causing the problems. And then, of course, on the other hand, right, you don't want too little immune functioning because that could set up a scenario like in HIV where you've got increased susceptibility to infectious disease or cancer. Not only the strength of the response is important, but also the appropriateness of the response. And so the immune system has to deal with a lot of different challenges. Some are extracellular, some are intracellular. So viruses are intracellular, right? Bacteria, most bacterial infections are extracellular. And so the immune system needs to develop a very specific response depending on what it's being challenged with. And so there's regulatory molecules and systems to set up those type of responses versus others. And if you wouldn't want a situation where you're mounting a response against an intracellular infection when really it's an extracellular infection. So it's highly regulated. And in addition, there's redundancy, right? So we talked a little bit about that. And redundancy means if you take away one component, some other component makes up for it, right? That's clearly true with the immune system. And like I said, it's diffuse in nature. It's throughout your whole body. And so by studying just blood, are you really knowing much about what might be going on in tissues? And it's sensitive to many different regulatory influences, which makes its study difficult. And historically, hardcore immunologists are reductionists, where they take cells out of a certain situation and put them in culture, right, in vitro, and then study the reactions of those cells in vitro. But, given my background and training, I'm more of a realist in that, is what's going on in that culture dish really what's going to be going on when the immune system is in its native state and being controlled by all those influencing signals I mean, we certainly can understand a lot by doing the reductionist approach here, but you always got to bring it back to the organism and try to make sure that what you're seeing in that reduced approach holds true in the system when it's all put back together. So some important questions in exercise immunology have been, how does exercise and physical activity influence immune function? Importantly, are these exercise-induced changes in immunity responsible for disease outcomes? And this is the big gorilla in the room, trying to figure out if a certain change in immunity induced by exercise is actually responsible for disease or some increase or decrease in susceptibility to infection. That's very hard to study and to prove. Importantly, what are the mechanisms of any changes that we see? Are there dose responses? And I think you'll get a good handle on that today when I go through this. And is timing important? I mean, timing is critical relative to how the immune system develops, and when exercise is applied, right, it may affect things differently depending on where in the immune system development the exercise is happening. So there's three major observations that I have kind of summarized that have driven our field to this point. One is the observation that when you exercise acutely, there's a very rapid and profound increase in the number of leukocytes in your blood. We'll talk about that. Secondly, in the 80s and 90s, there's some evidence that exercise, especially if it's intense and prolonged, might increase susceptibility to upper respiratory tract infections. That was another seminal finding, and we'll follow that up. And then lastly, probably a little bit more recently, we know that exercise can cause inflammation, especially if it's unaccustomed to you, right, muscle soreness and things like that. But there's certainly a literature that suggests that if you do exercise repeatedly, regular, moderate exercise training, that could have an anti-inflammatory effect. And so let's talk about that as well as the third observation. And again, I'll make a plug for this position statement. And as I was preparing this talk, I was like, wow, it's a revelation. Wow, I was involved in a paper 3 years ago that just did this, right? It's amazing how quickly you forget about what you did, a few years back in terms of your vita and your background. So let's talk about exercise-induced leukocytosis. So early on in the 70s, and even before that, studies demonstrated descriptively that exercise, even if brief, less than 1 minute, you could see an increase in the number of white blood cells very quickly in the blood. And then, of course, that finding got grabbed onto by a lot of researchers because it's very easy to do these studies. You can just take blood and then be able to look at these leukocytes in the blood. And there's a lot of phenotyping that went on and kinetic analysis to see, well, what cells are going up in the blood or any cells going down? What's the timing of these responses? And so to summarize a lot of work, I throw this up here not to confuse you, but here's cell number relative to time of exercise. So before exercise, leukocytes are at their normal level. When we do an acute exercise bout, these cell numbers rise, depending on what type of cell you're talking about. So B cells, B lymphocytes rise a little bit less than natural killer cells, than neutrophils and T cells and other cells. But you can see some of these responses are unique. In that, you can see neutrophils here in purple. Right? There's an initial peak right during and right after the exercise. Then there's a fall. And then there's a later sustained rise and then a fall. And this is out to about, again, it depends on the intensity and the duration of the exercise, but it could be up to words of 24 hours or so after the exercise. So you can see, in general, exercise causes a leukocytosis. But you'll also notice that in the post-exercise period, the concentrations of some cells go below what they were prior. Is there any significance to this in terms of the increase or the decrease? Those are important questions. So what are the mechanisms responsible for this increase? So there's been some work done to figure out, why does an acute exercise bout elevate leukocytes? One is demargination. We know that shear stress, the increase in blood flow and pressure, can basically knock immune cells off of the vascular endothelial cells. A lot of the immune cells are attached to the vascular endothelium, right? They may be wanting to get into that tissue or just kind of stuck there because there's counter-regulatory receptors on endothelial cells and immune cells that allow them to stick. They're pretty sticky, okay? So just by the fact when you start exercising, increase in blood flow and shear stress, like Mike mentioned, that can happen within seconds after exercise. We can see huge increases in the number of leukocytes, even before any hormonal signals or things could cause the leukocytosis. So clearly, shear stress has a role here. But there's also neuroendocrine mechanisms at play. Excuse me. The exercise-induced activation of the sympathetic nervous system HPA axis produces hormones, obviously, that bind to counter-receptors on immune cells that cause movement or even the production of immune cells in the bone marrow, hematopoiesis. We know that beta blockade can reduce exercise-induced leukocytosis. So that's some evidence to suggest that beta adrenergic receptors might be involved in this. And we also know cortisol can cause the bone marrow production and release of neutrophils. If you remember that slide I just showed you, there was an initial rise in neutrophils, then it went down, and then a second peak. Well, that second peak is due to the exercise-induced increase in cortisol, which stimulates the bone marrow to release some neutrophils into the blood. So diagrammatically, you can see these marginated leukocytes here shown in blue, marginating to the vascular endothelial cells of the blood vasculature. When we exercise, due to shear stress or interactions with these hormones, all of a sudden we get more leukocytes flowing in the blood, and if we sample that blood, then there'll be an increase in the count. So that's the increase in cells that we see during acute exercise. What's responsible for the reduction post-exercise? Well, it could be cell death. Some of those cells are dying in the post-exercise period, and there is some evidence to suggest that there's apoptosis or necrosis going on. The question is that a good or a bad thing? The jury's still out on that. We know that frank leukopenia is bad if you have low white blood cell count. But cell turnover could be a really good thing. One of the theories of aging is that your cells become senescent and their function is reduced. If exercise can increase the turnover of those cells and make new cells, right? In fact, there's a new literature on autophagy, the turnover of intracellular components, getting rid of the, I always think about getting rid of the old and bad and turning it into new, right? And exercise promotes that process. That could be a good thing. Another reason why you see these post-exercise reductions in leukocytes in the blood could be movement into tissues. And so, so Kruger in 2007 fluorescently labeled T cells and injected them into the vasculature of animals and then had the animals exercise intensely and basically looked to see in the post-exercise period where these cells ended up, tracking the cells. It's very important research because it then starts to speak to the mechanism. Well, where is exercise causing trafficking to tissues? What tissues, right? And so you can see here in these diagrams, this is cells per milligram of organ. So there's various organs like the thymus, liver, spleen, lymph nodes, bone marrow, Peyer's patches of the gut, blood, and lung. You can see in general that there's a reduction in the number of white blood cells in the spleen. So exercise actually causes contraction of the spleen and the release of many things, leukocytes being one of them. And you can also see in the post-exercise period there's an increase in tissues like the lung, blood, Peyer's patches, and bone marrow. So over here you can see the exercise-induced increase in these T cells in these different organs post-exercise. So knowing where these cells traffic to might give one clues as to what's going on, what the function might be of these cells, right? Or we could target a specific tissue and say, okay, well, now why are those T cells going there? What are they doing? Okay. So obviously the big question is, what's the physiological significance of these findings? We do know that leukocytosis is part of the fight or flight response to impair your body for impending wounds or damage. When you're exercising, you might be in a state where you need to defend yourself, right? So activating leukocytes in their movement might be important there. The question, though, is this a natural way to activate your defenses? And the jury's still out on that. This may have implications for health, but timing is important as changes are largely transient. And we know that if we look at somebody who's repeatedly or chronically exercised, somebody who's trained, they don't have any appreciable differences in their leukocyte counts or phenotypes in the blood relative to somebody who's untrained. Lastly, with this theme or thread, is there any way to use this response to our clinical advantage? And I've got a colleague at Houston, Ricky Simpson, who's actually demonstrated that acute exercise can increase the number of mature effector T lymphocytes in the blood. And it's these cells that clinicians are very interested in transplanting a lot of times. And so he's got a line of research where he's trying to convince clinicians, hey, we need to exercise these people before we take blood for these transplants because you're going to get more of the cells that you need to be able to do the transplants. And so that's kind of a cool application here, a clinical application. So just in conclusion, acute exercise transiently can mobilize leukocytes to the blood, part of the fight or flight response. Does this mean enhanced immune vigilance during exercise? We don't know. Some cells are reduced in the post-exercise period. Does that mean that there's a reduced immunity? We don't know, but we do know some of the tissues that those cells are trafficking to. And we know that exercise training has little effect on resting white blood cell counts, but that doesn't mean that immune cells are unaffected by exercise training, and I'm going to kind of get into that in a little bit. So the second thread or observation that's driven our field is increased incidence of respiratory tract infections after intense prolonged exercise. So the early findings by Dave Neiman, who's at App State, and many of you probably know David, indicated that strenuous prolonged exercise was associated with an increase in upper respiratory tract infections in the post-exercise period. So he did a lot of studies, including the kind of seminal work with the LA Marathon, that showed a significant increase, about a six-fold increase in upper respiratory tract infections in marathoners relative to matched controls in the week after the marathon race. So the observations like this kind of drove a lot of thinking, right? Oh, if that's the case, then what's going on, right? And it gave rise to this theory called the open window theory. So this open window theory is that high-intensity prolonged exercise might cause initial immune activation, but in the post-exercise period, this critical period between hours to days after exercise, right, we may be more susceptible to infection. So kind of like the open window, right? You open the window, stuff gets in, right, if your immune system is temporarily suppressed. But you'll also notice that if exercise is of low intensity or shorter in duration, that you get the benefit of the exercise without the post-exercise immunosuppression, so no open window. So it is these kind of theories that then drive the hypotheses behind a lot of the studies that have looked at exercise and immune responsiveness. To put it another way, right, you can formulate the J-shaped hypothesis, where if you're sedentary, you've got normal immune function or upper respiratory infection risk. If you're exercising moderately or training moderately, that risk is reduced because of an improvement in immune function. And then if you do heavy exertion or you overtrain or the stress of competition contributes to this, you actually get immunosuppression and you get elevated risk. And so this is very similar. I remember I actually gave a talk at UMass, and I was telling Patty about this. This guy, I can't remember his name, Calabrese, maybe? Calabrese, yeah. His whole idea was about hormesis. That which does not kill you makes you stronger. And there's a lot of examples in biology, radiation biology, where if you get a little bit of radiation, that actually is pretty good. But you get too much, that's bad. It's the same kind of theory here with exercise and some of our hypotheses related to immunity. So observations like that then led us to try to explain, what's going on with the immune system, right? Early work focused on exercise-induced changes in the function of cells called natural killer cells, which are cells that are responsible for defending against viral infections. They can recognize virally infected cells, and then they can lyse or kill virally infected cells. And in fact, we have an assay which we can utilize where we can take peripheral blood from people, isolate peripheral blood mononuclear cells, which contain natural killer cells and other cells, take those cells and incubate them with target cells, radio-labeled target cells, co-culture them, and then the natural killer cells will kill the tumor cells and release radioactivity into the supernatant, the fluid above the cells. We can sample that supernatant as an indication of cell killing. How effective were the NK cells at killing these tumor cells or these virally infected cells? And so an example of this is shown here. You can see on the y-axis is natural killer cell lytic activity in units of 25 to 1 ET, which means 25 effectors to 1 target. So you can mix various concentrations of NK cells or effector cells, the cells doing the thing, against the target cells. And what you see here in samples that were repeatedly taken during a 3-hour run or in the 21-hour recovery period is an increase in natural killer cell function or lytic activity during the exercise and a reduction post-exercise, and then ultimately kind of a leveling off. And so evidence like this tended to implicate, because the shape of the curve looked very similar to some of the other data, implicate NK cells in some causal way with changes in disease. However, there are interpretive problems of measuring immune function, especially from human blood samples obtained ex vivo. Like I said before, the numbers are changing, right? And so you could have increased numbers responsible for these changes in function, independent of whether or not exercise affects, importantly, function on a per-cell basis, okay? So there's interpretive problems here. Again, like I said before, all these assays are done ex vivo, and the cells are taken out of their natural environment. Is that really the way they're going to respond in vivo? And is blood really a window to the immune system? And I wanted to show you this to kind of give you an idea that, your leukocytes, your white blood cells are only, your white cells are only a fraction in the blood of relative to other tissues. So only about 2%, right? T cells are in the blood at any one time. About 2% of B cells. Natural killer cells, about 5%. Most of the cells are in other tissues, right? It's those tissues that we really need to be studying. So at the end of the day, we're really looking at correlational data between immune function and disease outcome. And then really the question is, can measuring anything in the blood really tell us about what goes on in the tissues in response to an infection? And so that shifted things to, well, let's look at the respiratory mucosa, because that's right at the interface between the outside and inside world. And of course, there are challenges that we face in our respiratory mucosa. Here's the respiratory epithelial barrier. These cells can secrete antibodies, in particular secretory IgA, as well as antimicrobial peptides. So they exist in the mucus layer that help prevent attachment and also neutralize viruses as well as bacteria. And to summarize a lot of work, acute bouts of moderate exercise have little impact on salivary IgA, but prolonged intense exercise or intensified training can evoke reductions in mucosal defenses that correlate with subsequent upper respiratory tract infections. A lot of this work is done by the Gleasons, who interestingly aren't married. One's in UK, one's in Australia. While this is a better measure of trying to explain upper respiratory tract infection, we only get correlational data between salivary IgA and the function. Time's up. All right. So a better approach would be to challenge people or animals with pathogens or immune activators. And this is work that's been done by me and some others. So we've taken a different tack. And, of course, challenging people with pathogens is probably outside of the scope of what the IRB would allow. But we've used vaccination responses, and others have too, as a way to look at whether or not exercise can protect us. And to make a long story short, we've shown that regular exercise training can improve antibody responses as measured by seroprotection in older adults. And these are advantages and disadvantages of this. And we're currently performing animal experiments to see if we can duplicate these findings to test mechanisms. So it's kind of like reverse engineering here. I can stop there. That's fine. I do want to acknowledge folks, people who in my lab who've contributed to this over the years and the funding. And sorry I went over, but you get passionate about your work. You get passionate. All right.